Okay, loop of Henle is the next section of the nephron that we need to sort of examine. So, we'll just draw ourselves a picture. Can't go down much there. No, I'll just bring that. I don't know what's happening. There we go. Now it's working. Okay, so loop of Henle. I need a sort of a downward loop here, so I'm going to move that up. So basically, we leave the proximal tubule, and the loop comes down. Now, there's something here at the bottom that I want you to be aware of. The bottom part of the ascending loop is thin, and then it widens to a thicker section. So make sure you show that it's widening up to a thicker section here. This is essentially the loop of Henle. This part is over here would be the proximal tubule. And over here would be the distal tubule. Okay, now, in red, we're going to write down some important things. This side of the loop of Henle has a membrane that is permeable to water. Permeable to water, but not salt. And the other side of the loop of Henle, which is on this side, is more permeable to salt, but not so permeable to water. Okay? In particular, the bottom section down here, actually, let's change that bracket. Let's just make it this sort of the bottom section. This part. This is permeable to salt, but not to water so much. Okay. The other thing we have to remember, and I'll do it with the shading, is that we have three sort of different layers, right? As the loop of Henle goes down, it moves through the cortex, right? And then underneath that, we had the sort of the outer medulla. What the heck just happened there? Undo, thank you. Okay, and this represents the outer medulla. And then we had the inner medulla in the sort of darky color here, the very dark orange. Okay? And so let's write those. Come on, pen. There we go. So the cortex, outer medulla, and the inner medulla. And do you remember what was important about this? What was important is the way the concentration changes. The concentrations in the tissues increase as you go down, increasing concentration. Let's put that somewhere on our little list here. I'll put it in a nice green color. So as we go down like this, we have increasing concentration. So what that means is the osmolarity increases as you go down. Osmolarity is sort of a measure of how much water wants to move by osmosis. So the more concentrated something gets, the more water will want to enter into that tissue. Yeah. Okay, so we'll pick another color. Maybe purple will stand out. So because this descending loop is permeable to water, that means water can easily move through it. And if you are dipping down through these tissues, which are increasing in concentrations, then obviously water is going to want to be pulled out of the filtrate, which is inside the tube. So in other words, we've pulled a lot of stuff out of the tube already in the proximal tubule, right? So the filtrate that's inside has less stuff in it. So as the filtrate moves down through here, what happens is water starts to passively transport out by osmosis. And of course, as the water comes out, the solution gets more concentrated, the filtrate, the urine, right? Yes, Lauren. Yes, it does. Okay. Now, as the filtrate is moving down the loop of Henle, down the descending loop, this side, the water's coming out, it's getting more concentrated. But as it moves down, the tissues are also getting more concentrated, right? The lower tissues, the outer and the inner medulla, are much more concentrated. So that means that even though we're pulling water out, as you go down the loop, you can keep pulling water out because the urine or the filtrate in the tube is still less concentrated than the surrounding tissue. So water is being pulled out all the way down this tube, and this is the reason why we need these three tissues to be slightly different concentrations, right? Otherwise, if you pulled water out at the top, then you'd have an isotonic situation. You'd have the urine filtrate in the tube the same concentration as the tissue, and nothing would move. But if the tissue keeps getting more concentrated, it'll keep pulling water out because the osmolarity increases as we go down. And that's what you have to understand. Okay, now we spin around the bottom. So what we basically are doing here is concentrating the urine, and we're saving water, right? We're bringing water back. Okay, now in the ascending loop, which is what this part is called, there's two sections. There's the thin part and there's the thick part, all right? Now this has a different permeability. This side of the loop is permeable to salt, but not to water. So the cell membranes here are slightly different, right? They have a slightly different structure. I should have this on. So as the as the filtrate comes down the descending loop and this water comes out, what's at the bottom of the loop is a very concentrated filtrate, right? Very dense and lots of salts. And so as the permeability changes from salt, or from water to salt, we start to see salt moving out on this side. So let me pick another color, maybe blue. We start to see salts moving out passively, NaCl. Because the permeability has changed, and don't forget, this inner medulla is very salty, right? But the concentration of the urine is even saltier because of all that water that's been pulled out. And now the salt sort of starts to move. And this is what contributes to the saltiness of the inner medulla. This is the mechanism that keeps it saltier than the upper tissues in the kidney in this picture. But it's all because of the permeability change of those membranes. That's kind of the key, the membranes in this distal tubule. All right. And then as it ascends the tube, once you get up into the thick part, everything so far has been basically passive transport. I don't know how we can indicate that on the picture. You might indicate it with words on your picture somehow, but all of this is happening passively due to the differing concentration gradients. But then in the upper part of this, the kidney will now, how about I use a dash line for active transport? Maybe that'll work. Active, and we can make a little key later. Salts are now actively transported out of here. The reason they have to be actively transported out is because as the salt leaves, the solution now becomes more diluted, right? You've got to keep track of the solutes and how they move. So at first, it becomes very concentrated. Then, as the salt comes out, it starts to get a little less concentrated again, but you're also moving back up where the tissues are less concentrated. So in order to get this to do its job, we have to actively transport the sodium out here, which then makes the sort of filtrate less salty and more dilute. So this entire loop of Henle is basically the way we get water out of the urine, the way we can take a very, very dilute urine and we can save water. And the mechanism that makes it work is the differing permeabilities between the descending and the ascending loop. And the other very important thing is the increasing concentration in the tissues of the kidney as we go down towards the inner medulla, which allow for this automatic thing to happen. So there's the loop of Henle, and that's essentially what's going on there. Question? Okay, so the question is, because the concentration is greater at the bottom, does more water come out? Not really, because you have to think of the two things together. In fact, what I can move down here and draw you a little thing. So here's the descending loop, right? And if I just put dashed lines to show the tissues, remember that osmosis is all about comparing to your surroundings. So at first, you've got dilute, right? But the tissues themselves are not so concentrated either. So because it's dilute and the tissues aren't concentrated, not much happens. It's kind of like an isotonic situation, right? The concentration of the tissues is the same as what's in the tube, so no big deal. But as you go down the tube, this is more concentrated. What that means is it's essentially saltier. So that means that as the solution moves down, there is now an increase in osmolarity. There's now a difference in concentration. And because salt can't move through this membrane, it causes osmosis. It causes water to come out. This concentrates the urine. As you go down further, now the urine is more concentrated, right? So now you have concentrated urine. So if this was the same, it would reach an sort of an isotonic situation again, and nothing would happen. But this is even more concentrated now, even more, which means that as this flows down, more water can come out. So it's not so much that the rate of water moving is changing. It's just water continuously is moving out of the tubule because the tissues are always slightly more concentrated than what's inside in the filtrate. Okay, that's a good question. And then on the opposite side, since we're talking about it like this, on the opposite side, it's the opposite because the permeability is opposite. On this side, salts can't move. And remember from your grade 9 and 10, if salts can't move, then water will move by osmosis, right, to try to even out the gradient. But on this side, the salts can move. And the water doesn't move so much. This membrane is more impermeable to water. So on the way back up, instead of undoing everything that's been done, on the way back up, actual salt actually leaves the tubule, which then dilutes the filtrate a little bit more. So in a sense, we've taken this very large volume of filtrate. It's got a large volume because in the proximal tubule, everything's come out of the bloodstream. And we've concentrated the solution first by withdrawing the water, and then we now are withdrawing the salts. So we've taken a large volume of solution and whittled it down to a small volume of solution. So when the salts come out, and they're actively transported out at the top, so what we end up with is a volume reduction. That's an important thing, a volume reduction. And we've conserved water by doing that. So we're actually going to be making less urine and not have the same volume. Okay, let's move on then to the distal tubule, which is sort of the second last section. And I don't know how to move that back onto the other side where it belongs. So now we'll talk about the distal tubule. So you come up from the loop of Henle, and now you have this wiggly sort of tube, right? Distal tubule. And we already mentioned pretty much what happens here. Essentially, we have the regulation of sodium ions. Sodium is a very important, or sorry, potassium, I mean. Potassium is a very important thing to regulate. And so if you, typically what's happening is potassium, remember, is being actively transported in. H plus is being actively transported in. Salt is being actively transported out, NaCl. And bicarbonate is being actively transported out. And then there's a passive transport of H2O that tends to happen because of the movement of these ions. It's very similar in the proximal tubule. What's really going on here, though, for the most part, is it's the recovery and management of some electrolyte balance, but also the pH. The bicarbonate and the H plus is monitoring the pH. So let's write down pH regulation. And we can also put electrolyte regulation. Electrolytes are the words that mean any of the charged ions. And some of them are also the pH ions, but we'll just kind of put the two together. So that's really all we need to say about the distal tubule. So what we've got now is from the Bowman's capsule, we had this large outflux of stuff from the bloodstream. We've pulled back the nutrients, we've managed the electrolytes, we've managed the acidity of it, we've sent it down the loop of Henle, we've pulled out a lot of water, and then back the way up, we pulled out a lot of salt, we reduced the volume so that we're not urinating 20,000 gallons of fluid a day, and we're conserving water, and then we now have reached the collecting duct. And in the collecting duct, there's a couple more things that are going to take place. The distal tubule has no variations in thickness. It's just regular, yeah. So this would be the collecting duct, which comes after. And there is an interesting little thing that happens here, because the collecting duct also travels through the three layers. Instead of the colors, I'm just going to take this sort of brown color here and just do some dotted lines. So we have the, right, these are the three tissue layers, the cortex, the inner medulla, and the outer medulla. And it also passes through here. So it can do a few little things in the collecting duct. As the filtrate is moving down here like this is the place where even more water can be captured. So the filtrate is coming down. It's relatively watery again, isn't it? And so as it moves down the collecting duct, we see the water will be coming out all the way down here because the collecting duct is moving again through more and more concentrated tissues as it moves down. Here's the interesting thing about this, though. The collecting duct permeability is hormonally regulated. The permeability, ability of the collecting duct, I'll just call it the CD for now, of the collecting duct is hormonally regulated. In other words, hormones can change the permeability of this. So what that means is that the degree of water that's reclaimed can be controlled through hormones. We'll talk about hormones later. So this is where basically your body decides how much water you have available and how much it wants to waste in the urine. And it can make the urine more concentrated or less concentrated depending on your hydration state. Okay? And so when you go to the bathroom and you pee really dark concentrated urine because you haven't had water in a while, that's because the collecting duct is sucking as much water as it can out of here because you are potentially dehydrated. But if you're well hydrated and there's lots of water available, that will change the concentrations of these outside tissues slightly, and less water will need to be pulled out, which means your urine can be a little more watery. So that's one of the things that's happening here, is the hormonal control of how much water is actually reduced in the urine. Okay, the permeability of the collecting duct is pretty much just water, but near the bottom of the duct, we also get some outflux of urea. Now you might think that's weird because the urea is the waste product that we're trying to get rid of, right? But by pumping the urea out here, or actually by having it pass out here and having it permeable, this contributes to the higher concentration of the inner medulla. That's what keeps it so osmotically high in terms of its concentrations. So what the kidney's doing is it's using its own waste to pump it into that area to keep the osmotic pressure high, keep it salty, right? So why waste salt? We have this urea kicking around. If you put any solute in there, it's going to change the concentration. So the urea is moving out here. It's a passive movement because the membrane is slightly permeable to urea, and it allows it to move out down here, and this contributes to keeping that bottom section, that inner medulla, very, very salty, so that it can do its job in the loop of Henle. So in a sense, we're not reclaiming this urea. It's not going out into the bloodstream and taking off back through the body. It's just hanging out in this inner medulla to help it stay very salty. Question? All right, we're using this waste product of urea to help keep the concentrations where we want them to keep the kidney functioning. Anybody ever eaten kidney or cooked kidney? When you cook kidney, what do you smell? Urine. And that's because the actual tissues of the kidney contain urea, because that's something that the collecting duct is putting in there to make those different concentrations, right? So, yeah, kidney. Yum. Okay, so that basically ends the story. So if we were to sum up, very quickly everything so far, if we take the glomerulus and the Bowman's capsule, remember the glomerulus is the term for the big ball of capillaries, and the Bowman's capsule is the term for the shape of the tubule, the mouth shape. So what happens there is basically blood pressure drives everything out of the blood. Drives out everything. Now, I'm going to put everything in quotes. And by everything, I mean the things that we talked about, right? Everything. Blood pressure drives everything out of blood into the tubule. This makes for a large volume. So this makes a large volume of filtrate. When you've got all these little guys working together, remember there are up to 1 million of these in the kidney. They're all working together, sucking stuff out of the blood. Okay, so that's what's happening there basically. Then, yeah, I'll go back up. Then, we move on to the proximal tubule. And we've already kind of summarized that one in the last video, but I'll just write it again. So for the proximal tubule, we have three sort of things going on. One is the reclaiming of nutrients. The things that were pushed into the tubule that we want to keep. So those get pulled back. That's what I mean by nutrients, right? Sugars, anything like that that's good for the body type of thing, okay? The reclaiming of nutrients. the balance, or at least helping, the proximal and distal tubule work together on this. You'll notice how they're both in the same cortex, right? They're both in the same tissue. So anything that one pulls out, the other pulls in, they kind of work together. So they balance pH by the movement of bicarbonate and hydrogen ions. Now lastly, there is some movement of salts in water. But there's really no major net effect of this, because you're pulling salts out, water's coming out, following the salts, which means that the concentrations aren't actually changing much. So there's some movement of salt in water, but little net effect, is what I'm going to say. Little net effect on, effect on osmolarity. So it doesn't change the concentration enough to cause something to happen yet. But when we get to the loop of Henle, that's where the osmolarity becomes the key feature. Question. Okay, we move down. We move down to the loop of Henle. And it sort of has it has the, let's call it the descending side, the descending loop. What's happening here is water reclamation. Water is being pulled in, and it's due to the increasing osmolarity of the tissues around. Okay? Let's put due to. Let's just put osmolarity. Yes. Remember, out means it's coming back to you. If it stays in the tube, it's going to be urine and it's going to be wasted. So anything that comes out of the tube is reclaimed by the body through the bloodstream. Okay? That's what you have to remember. And then secondly, we have the ascending part. Ascending. Okay, let me just do it like this. And then we have salt movement, both active and passive. Maybe we'll put passive and active. Salt movement, passive and active. Oops. Which well, actually, why don't we put it like this? So both of these things are combining to reduce filtrate volume. Right, we're reducing the filtrate volume, and also let's put reclaiming water. I'll get a smaller. Reclaiming water, to some degree anyway. And then lastly, we have, well, not lastly, we have two things left. So let's go on to the distal tubule. And the distal tubule is kind of continuing the work of the proximal tubule. Because of all the movements of salts, things might be a little bit out of whack in terms of osmolarity again and so on. So the distal tubule is, again, regulating pH, making sure that after all this in and out of water and salts, that the pH is still where it needs to be. And that's essentially the distal tubule's job. And there's some electrolyte balance as well that we said. And then lastly, the collecting duct. The collecting duct, of course, sounds like just, oh, it's just collecting stuff, but it is not. It's actually doing two very important things. The collecting duct does, well, I mean, functionally, it collects the urine and sends it to the renal pelvis, which then directs it into the ureter and down to the bladder. So its job is to collect, but it also does some very important osmotic things, and one of them is water reclamation again. And the important thing here is to remember that this permeability is hormonally controlled. So the body can make decisions as to how much water is put into the urine. This is not because of differing concentrations anymore, and it's not just a passive thing that's happening in the tubule. What's happening here is the body saying we need to conserve water, so our hormones are going to shut down the permeability of this collecting duct to water. One of the things that you may have heard of, a diuretic. I can just put this here. It's sort of not part of the summary, but it's kind of interesting. A diuretic is a chemical that will make the collecting duct very permeable to water. It increases the permeability of the collecting duct, which means, I'm sorry, no, I've got that backwards. It decreases the permeability of the collecting duct significantly, which means water stays in the duct instead of coming into the tissues. There's no reclaiming of water, which leads to very dilute urine. And so many of the things we eat and drink are diuretics because if you keep water in the filtrate, it increases the volume of your urine and you pee a lot. So caffeine has been known to do this. Alcohol can have this effect. Various things that we eat and drink, many different kinds of drugs can have this effect. So what they do is if I go back there for a second, just to the picture, we said how water was coming out here, right here. The drugs can change the permeability of the collecting duct so that water is not allowed out, and the water stays in here, which then goes out into the urine and becomes very watery, and you tend to have a higher volume of urine, and it's a low concentration, so you're peeing a lot. And that can dehydrate you eventually because those drugs are not being regulated by your own body. They're just a drug that you've taken, right? So you have to be careful. This is why when you drink alcohol, you tend to pee a lot and you become dehydrated after because your kidney isn't working the way it's supposed to. It's not able to reclaim that water. So water reclamation, and the other thing is the utilization of urea to maintain higher concentrations in the medulla tissue. In the medulla tissue. And remember, those higher concentrations are important. If those concentrations aren't maintained, the loop of Henle doesn't work. And so that's what the reclamation of this little bit of urea into the tissues of the medulla helps keep that concentration very high and increases the osmolarity, right? The movement.